USA, Saudi Arabia, and Germany. He has a track record of more than 100 international publications, 10 patents. He served as a referee and editorial board member for many international organizations and Q1 scientific journals, and supervised more than 100 master and doctoral theses from 2011 to 2016. He served as the Dean of Pharmacy at German University in Cairo and Ambassador Scientific to Alexander von Humboldt Foundation, Germany in Egypt. Dr. Ashraf also is a member of the Drug National Committee, ASRT. Welcome, Dr. Ashraf, and we are listening for your...
Okay, thank you so much, Professor Tariq, for the kind introduction. And of course, my pleasure to have as a chairperson Dr. Nadia. She's the head of the National Drug Committee, and of course, my colleague, Dr. Mustafa Al-Arabi. Most welcome. Okay, today I'm going to brief you about a research that has been done in my group. These are the outline of the presentation. For the introduction, Spirooxindole are unique chemical scaffolds and reported so many times in many active compounds. So, as you know, spiro compounds are those in which two rings are made together by one atom. And there are so many reported Spirooxindole compounds. As you can see here, the indole is linked to the other ring. Either it is a carbocycle or a heterocycle, and my main focus will be compound Mi888. These Spirooxindole derivatives, they have stereochemical restriction around the spirocenter, and this gives a unique 3D structure and the structural complexity and rigidification that plays a crucial role in proper binding to a particular biological target. We have so many compounds as pyrrolidino Spirooxindole. We have so many compounds reported in literature with different types of activities. And most important for us are those three compounds with the codes MIs. These compounds were found to be of potent anti-cancer effect, particularly against colorectal cancer, and their reported mechanism of action is inhibiting a protein-protein interaction between p53, which is the guardian of the genome, and murine double minute 2 protein, which is a negative regulator of p53. As you can see, all of these compounds are pyrrolidino-oxindole derivative. p53 is known as guardian of the genome, and it is activated in response to many stress factors like DNA damage, oncogene signaling, hypoxia, and so on, and it exerts its apoptotic effect and cell cycle arrest or DNA repair almost by 18 different mechanisms. Here you can see how p53 is interacting with MDM2. Most important for us is what is known as the three hot spot interactions. So the interaction between p53 and MDM2 is mainly dependent on the presence of phenylalanine, tryptophan, and leucine, which are present in the deep cleft or the deep pocket of MDM2. And of course, if you can block such amino acids or prevent the interaction with these amino acids, in this case, you are going to inhibit MDM2 and spare p53. Here you can see some of the compounds reported as MDM2 inhibitors. Here you can see this compound. This compound Mi77301 is actually currently undergoing phase 1 and successful, and then phase 2 for treatment of colorectal cancer. So it is now in clinical trial. As you can see here, we have pyrrolidine attached to oxindole, and in addition to some aromatic substituents on the pyrrolidine part and other substituent on carbon 2. It is proposed that this compound is inhibiting MDM2. Mainly this indole part is similar to the indole of tryptophan, which is like one of the 3 hotspots of MDM2. And these 2 substituents with the red cycle are mimicking the interaction with leucine 26 and phenylalanine 19. So actually this compound can block the hotspots of MDM2, sparing P53. For our part, we'd like to prepare pyrrolidino oxindole derivatives mimicking the MI series of compounds and to test their in vitro activity as potential anti-cancer agent. Of course, by testing their growth inhibitory effect against many cancer cell lines. In our work, it is the one upstairs, it is the one to the up right side up. As you can see, these are like the reference compounds. So here we have many changes. We keep the structural spiropyrrolidinyl oxindole core nucleus. However, here we have changed the position of the pyrrolidine nitrogen linking to the oxindole, so it is now in position 2 rather than position 3 in the reference compounds. We have changed the indole ring a little bit by substitution by a halogen, and this halogen will be in position 6 rather than position 5 in the reference compounds. Also, we introduced 2 diaryl substituents, and they are introduced in order to mimic those diaryl substituents present also in compound like natlin 3, which is known as a very potent MDM2 protein. For ring A, actually we introduced so many substituents. So, for example, we used the chlorophenyl, 4-chloro, 2-chloro. Here you can see the nature of the different substituents we introduced on ring A. And for the halogen, so here we have at 5 position, 5-chloro, 5-bromo, and we have the unsubstituted oxindole. For ring B, we have this hydroxyphenyl, chloro, and alkylated one. For synthetic pathway, mainly we have a reaction between acetophenone and aldehyde to give the corresponding shell cone. This is followed by a multi-component reaction with the desired isatine and benzylamines to give the spirooxindole compounds. By this, we have prepared 22 compounds by this scheme. The importance of this scheme from chemical perspective is that it includes a multi-component reaction, which is like a tool that can serve green chemistry. So instead of sequential reaction, here we have multi-component reaction. For scheme 2, in this case, we started with hydroxyacetophenone with an appropriate benzaldehyde derivative. This is followed by alkylation of the OH, followed by the same reaction, and in this case we obtain the alkylated derivative or alkylated derivative, and by this we obtain 6-pyrrolidino oxindole. The structural elucidation, of course, I will not expose this part, but it is done by LCMS 1H NMR, carbon 13, and 2D NMR tools. For biological compounds, for biological testing, 27, almost all the synthesized compounds were tested for their growth inhibitory effect on a panel of 60 human cancer cell lines, representing 9 panels in the National Cancer Institute in the United States. Those 60 cancer cell lines are representing 9 tissue subtypes, including leukemia, non-small cell lung cancer, melanoma, colon cancer, ovarian, CNS, renal, prostate, and breast cancer. And the primary screening is done at 10 micromolar concentration against these 60 cell lines. For the good results, 11 of our compounds showed very potent growth inhibition to these cancer cell lines, and it was above 100% for those 11 compounds. This means that the mean GI50 is expected to be less than 10 micromolar. So as you can see, we have very potent compounds in growth inhibition percentage at 10 micromolar. And 9 compounds showed growth inhibition between 50 to 100%, again against all these 60 cancer cell lines. So these are the mean values. And 17 of the tested compound were escalated for the second testing, which is a 5-dose testing. And in this case, the compounds are tested for growth inhibitory effect at concentration range ranging from 100 micromolar to as low as 10 nanomolar. So these are the tested compound in the second testing. Here we can see a clear structure-activity relationship. For example, this compound with on ring B, we have 2-ethoxyphenyl. As on ring A, we have 2-ethoxyphenyl, and here we have para-ethoxy. We can find that the mean GI50 on the 60 cancer cell line is better with the 2-ethoxy compound. This might be due to its effect on the conformation of the molecule. Another, for ring A, we have like methyl ethoxy and the chloro substituent. Again, the 2-methyl substituent, 2-methylphenyl, looks as the best compound with mean GI50 against the 60 cancer cell line of less than 5 micromolar. For 1J, it's 11. For 1E, it was 17 micromolar. So they are very potent, and actually this is the mean of the activity against the 60 cancer cell lines. For the indole halogens, as you can see, the best activity was obtained with the 5-bromo indole, followed by the unsubstituted, followed by the chloro, and this may arise the chance or the possibility of having a halogen bonding between the bromo and the receptor. For the free OH versus alkylated compounds, the alkylated compounds looks to be much more active than the non-alkylated one. For example, compound 3C, its mean GI50 is 2.5 micromolar. The other alkylated one is 3 micromolar, and for the non-alkylated one, it is 13 micromolar. So here you can see in order the activity of those 11 compounds. They are mean GI50. It ranges from 2.5 to 17 micromolar against the 60 cancer cell lines. And this is like, you can say it's very potent compounds against those 60 cell lines. We tried to see if there is a sort of selectivity against a particular cell line. So we calculated the mean GI50 to each particular panel, leukemia and non-small cell and so on. And we compared this with the mean GI50 against the 60 cancer cell lines. If we divide the mean GI50 by the leukemia, for example, or any particular panel cell line, and it is 3 to 6, it means moderate selectivity. If more than 6, it is high selectivity. But what we obtained is like broad spectrum growth inhibitory agents. There is no particular selectivity to any panel at all. But you can see here, for example, leukemia for compound 1. We have here the best cell line. I can see that it is non-small cell lung cancer for compound 1L. So from this slide you can know which panel is the most sensitive to these compounds. In conclusion, 27 novel compounds with variously substituted Spirooxindole core nucleus were synthesized, structurally elucidated, and evaluated against as growth inhibitory agent against 60 human cancer cell lines representing 9 panel types. 11 compounds showed mean growth inhibition more than 100% at 10 micromolar, with compound 3A showing the highest inhibitory effect. It is 180-50%, and actually this is a compound with the O-alkylated oxygen, or with the oxygen alkylated. 17 of our tested compounds satisfied the primary screening threshold and escalated for 5 dose testing, and they showed mean GI50s ranging from 2.5 to 17 micromolar, indicating their high potency as growth inhibitory agents. There is no selectivity towards a particular panel, and the compounds can be described as broad spectrum growth inhibitory agents. I would like to acknowledge the contributor of this work from my group. So this work is mainly abstracted from the master thesis of Toqa Abdelgaber. Dr. Khaled was her examiner, and with Dr. Marwa Hani, she's a co-supervisor with me. And of course, I would like to thank the National Cancer Institute for carrying out all the growth inhibitory testing. In final, I would like to thank you for your time and good listening.